Well, that's a great start. So when I saw Robin yesterday, I said, Robin, whatever you do tomorrow, enjoy yourself. Are you enjoying it?
Yes, thank you.
Good, excellent. Good. So this brief presentation is going to be in two parts. And the first part is going to be about Robin and me. Is this working? Yeah. And in particular, how Robin influenced my thinking and subsequent and career, therefore. And the second part is about assortative mating, which to my knowledge has absolutely nothing to do with Robin, but I thought it was fun to talk about it. Some unpublished work. That's Robin. Okay. So Robin's influence on my, on my research and subsequent career, well, the obvious starting point, as everybody else has been talking about, that is, of course, REML. And in particular, that it's actually a very obvious and natural way to estimate variance components. And as Robin taught me many years ago, because for a balanced design, it just collapses to an analysis of variance, which is how Robin's always thought about it. Say yes?
Yes.
Yeah, okay. Good. And also, what I come back to later is thinking actually about within family segregation variance. That comes in some of our work from the past as well. Decomposition and partitioning has been mentioned on a number of occasions, and that's been a great influence on my subsequent work. And finally, Robin's, I'm sure he's made this quote in the pub sometime, that all models are wrong, but a mixed linear model will do a job most of the time. Okay, well, this is from my paper from my thesis, and it just shows you, this is just an example, without going into the details, that we're already starting to think about partitioning of variation. This was just an application to dairy cattle, where we tried to partition additive genetic variation in component due to Mendelian sampling terms in males and females. It's just an example. And you've seen part of this picture before, but not a complete picture before. This was at a time when we were young. We had hair. This is our chair. You might not recognize him. That's Robin at his this is your DSC party. That is Sarah Knott over there, who looked so young that every time we went to the pub, they were asking for an ID. And she was 26, I think, at the time. And this is an Irish gangster called John Ruan. I have no idea who that chap is. Okay, so moving on a few years, this is actually when I was working with both Chris and Robin at Roslin. And I'm not going to go through this horrendously looking equation, but it's basically starting to think about models where you have many, many segregating loci that are linked, and looking at within family variation, because this was infinitesimal type model where you had variance segregating in a backcross population. And this was just to do with marker-assisted introgression, but at the same time we're also thinking about how many genetic markers would you actually need to capture that segregation variance. And that came back later in my work in human genetics. So my first foray in human genetics was actually because of encouragement from Chris at the time. There was a paper published by colleagues in Edinburgh in psychiatric genetics, and who had for human geneticists, a very large pedigree of I don't know, about 150 individuals. That was large, and had done some linkage analysis to try to find genes that were associated within families, so linked to bipolar disorder and psychiatric disorder. And I thought, well, we can actually apply a more quantitative, take a more quantitative approach to that and do a linkage analysis with variance components. And of course, I wanted to apply REML. So this was we started this in 96 or 97. And to my knowledge, there was no program off the shelf available to actually run this, because we had to have both a pedigree and A matrix, but also a user-specified locus-specific A matrix, and realized a relationship that was estimated from markers. So I just looked up at one of the equations, which I'm sure I got from Robin, and I actually, because it was only a pedigree of 200, say, with the same number of individuals as records. So I just basically programmed this. This is probably the most inefficient REML algorithm ever programmed, but I just programmed it on the V matrix at the time. I used REML because, I was taught that's what you do to estimate variance component. And as Chris mentioned, that's now quite popular in human genetics. But really, whether you use REML or ML doesn't make one iota of difference, because the degrees of freedom we usually have in human genetics is very, very small compared to the number of observations. But still, it's a matter of principle. We also use MCMC to estimate identity by descent at the time, using a method or program from Simon Heath, who was also in Edinburgh, or had been in Edinburgh. And I reckon that this, both REML and IBD and MCMC for IBD was a bit too novel for the field, because this has racked up 36 citations in total, and it was an awful lot of work. But these are just some of the examples which kind of led, this thinking at the time led to subsequent work where we estimated with sufficient data and genetic markers, estimated variation, segregating variation in families, and also variation that's captured by common SNP variants. And I'm not really going to go into that in great detail. So I had GREML in the title, and Chris already mentioned it. What not a lot of people know is where the term actually comes from. It was, to my knowledge, it was first suggested by an economist called Daniel Benjamin, who I still collaborate with, where we wrote a paper on the genetic architecture of economic and political preferences. There you go, REML has been applied to estimate genetic variance associated with economic and political preferences. This was from 2012, I think. So GREML is implemented in a software package written and maintained by my colleague Jiang Yang at the University of Queensland. It's actually based mostly on an AI algorithm, but also on, it has a little bit of EM to get going. Because of the data structures in humans, that the number of individuals is typically similar to the total number of observations, we don't work with the mixed model equations at all. So you might think this is very inefficient, but it runs pretty fast, because I heard yesterday from Gail Davis, who's in the audience, that she's run data sets of the order of 150,000 individuals, so this is a V matrix of 150,000, and estimated multiple variance components, and that just takes a few days. And at the moment we have some implementation to make it a lot faster, but that's not released yet. Okay, well that was the first part. Now the second part, which I don't think has anything to do with Robin, although I did notice that in your 76 paper, where you show how to analyze data on parents and offspring, you mentioned that that's suitable for assortative mating as well. So assortative mating is just as most of you will know, it's a form of non-random mating that is characterized by phenotypic similarity between, in this case, spouses or parents. And exemplified here by this fatal attraction here. You could think of what the phenotype is, whether it's nerds, whether it's height, or whether it's the color of their clothes, but it's definitely assortative. So why are partners, when you look at, I'm just thinking about human data now, why are partners or spouses the same phenotypically, or why would they be similar? What's possible reasons for that? Well, one is that because they meet each other in a specific sort of social environment or cultural environment, so social stratification in the population. Another one is they get together and have some convergence over time. For example, they have similar diet and what have you. But there's also, of course, the one which I think is genetically and from an evolutionary point of view most interesting, is that they are similar because essentially the mate choice was driven by the phenotypic similarity. There's a big caveat there, and Douglas Faulkner already mentioned it in the first edition of his book. Assortative mating, this should be, I typed this last night, that's a typo. In man, however, probably seldom arises purely in this way, by which he meant phenotypic resemblance is the cause of assortative mating, and caution is needed in applying the results to human data. So we now have big data also in humans. For example, the UK Biobank. It's a data set of 500,000 individuals for which we have SNP data that's imputed to a fully sequenced reference, and we have many, many, many phenotypes. And as I'll show, you can exploit the availability of these data to ask questions that had been clearly raised before, but to actually try to answer them with novel data. But before that, I go back to Visscher 18, because that was mentioned by Augustine. It's a very notoriously difficult paper to read, as Augustine mentioned. But what a lot of people don't know is the majority of this paper is about assortative mating. It starts, here you go, it starts here at page 27. And Visscher later commented on the fact that he thought that of all the theory that is in this paper, dealing with assortative mating was the most complicated. A few years later, there's another famous paper for Sewell Wright, which he used these path diagrams, which I find absolutely impossible to deal with, but others don't. And interestingly, at that time, I don't know whether the post hadn't arrived yet, but Visscher 18 is not cited in this paper from Sewell Wright. Visscher dealt much more with what we now would say almost an infinitesimal model, many loci, and a lot of Wright's derivations was with finite locus models, particularly a few loci. And in human genetics, the sort of the group that have mostly looked in the past at assortative mating before the advent of DNA markers were twin researchers. And I would say, unfortunately, they also inherited Sewell Wright's way of modeling things. So you can tell me what the model is here. I can't work it out, but this is what the twin researchers work with, structural equation modeling, and this is a paper from Andrew Heath and Lyndon Eves in 85. So what are the genetic consequences of assortative mating, of like with like assortative mating? Well, it creates essentially linkage disequilibrium in the population, but it's between also unlinked loci, or mostly between unlinked loci, and it's properly phrased as gametic phase disequilibrium. So compared to a randomly mating population, we're going to have a positive correlation in the, I've called it genetic values, but to you it's just the breeding values between mates. It increases genetic variance in the population relative to random mating. It increases homozygosity of trait loci, but this is not a really big effect when you have very polygenic traits. And it also increases the correlation between relatives. So in the past, the correlation between relatives has been studied for very close relatives. But the biggest effect of assortative mating is actually an increase in the correlation relative to random mating of distant relatives. And I've tried to show that here. So here we have relatedness on the x-axis. On the right-hand side, monozygotic twins, and on the left all the way to relative pairs that have a relationship of 1 in 1,000, or 1,024. And here's an example of unilineal relatives, and here of the descendants of full SIBs. And the red bars here are representing a trait like human height with a high heritability and a phenotypic correlation, which is this parameter rho here of about 0.2. And the blue one, something more like how many years of education you've had. With a lower heritability, or this might be a bit too low, and a higher phenotypic correlation. And as you can see, if you just focus here on these distant relatives, that the increase in their, although the absolute correlation between these relatives is very low, the relative one is of the order of 2 to 4 for these distant relatives. And in principle, we now might have actually data to detect that departure from a simple additive model where you don't have assortative mating. So just to sort of finish off in the last few slides, we looked at this, and others have looked at this as well, but I'll just show you results from our group. We looked at this in two ways. First of all, if you ascertain if you have spouse pairs with phenotype data and with SNP data, you can essentially look at an association between the phenotype of one individual and the genotype or the breeding value of their spouse, or the estimated breeding value. That's one way of looking at the data. But a much more fun way, I think, of the data is that we know from theory that we're getting a predicted effect of gametic phase disequilibrium in the population. Can we actually go into the population knowing something about which loci are associated with a trait of interest, and can we detect an imprint of assortative mating by looking at by trying to get a handle at gametic phase disequilibrium? So the first one was actually published last year, where we took a number of data sets. Is this running out of battery or not? No. That's shown over here. Here we have a bunch of different data sets, and here I've just shown you just a simple phenotypic correlation of the spouse pairs. We had about 20,000 spouse pairs here. And here on the right side, we've basically done a regression of the phenotype of one individual on their estimated breeding value of their spouse. And under some assumptions that a regression coefficient is the same as this phenotypic correlation in expectation if the reason for the correlation is essentially the choice of phenotype when they get together. So we call it deliberate assortative mating for height. And whether we looked at a male as the phenotype and then the estimated breeding value of their female spouse partner or the other way around, we're basically getting the same result. And when you look at the average, it's bang right, very, very similar to the observed phenotypic correlation, suggesting but not proving that indeed there is assortative mating and mate choice based upon height. The other one is looking in the population as such and looking for an imprint of assortative mating. So what we did here, we went to the UK Biobank and basically treated it and a few other data sets as just a sample of genomes in the population. So we knew from outside these data that we have loci in the genome that are associated with a trait like height or educational attainment and a bunch of other traits. And we basically looked for whether or not within a person there is a correlation with how many increasing alleles you have on the odd chromosomes and the number of trait increasing alleles you have on the even chromosomes. And that is a measure of gametic phase disequilibrium. So that's our metric that we used. And when we did that, and we had a theoretical expectation of that from knowing the phenotypic correlation of the spouses and how much genetic variance was captured by the loci that we looked at. And here the results on the left-hand side for height, where our estimate of gametic phase disequilibrium, which is this correlation, is about 0.03 from these different data sets. And that's exactly what we predicted beforehand from very few parameters that we had available to us. And the second one is similar, is for educational attainment, where we also get a correspondence between the theoretical prediction and the results and the theory. And we took a null trait, bone mineral density, which is a risk factor for or a predictor for fractures, where we didn't expect there to be any gametic phase disequilibrium or any correlation within individuals of how many increasing alleles they have on the odd chromosomes and the even chromosomes. And for that, we didn't find anything. Then subsequently, we went to because in between us doing this and this analysis, we received the full UK Biobank data, and we could just pick out, just from the UK Biobank, about 20,000 spouse pairs. So we derived a theoretical relationship between the correlation in the EBVs between the mates and our estimate of gametic phase disequilibrium, all based on exactly the same SNPs. And we looked at about 30-odd traits, and we found for two of them that the estimates are very close to the theoretical expectation, and again, that's height and educational attainment. And for other traits, which include, for example, liability to psychiatric disorders, we don't find evidence here, but this is partly a power issue, because for those specific traits, like liability schizophrenia, the genetic predictors still don't explain enough genetic variation to get these guys off the line here. So maybe in the future we get evidence for assortative mating on liability psychiatric disorders. So this is, before my last slide, this is our group in Brisbane. And I just want to point out that not only are there a lot of academic grandchildren from Robin, so that means students and postdoc of his students, there are also great-grand offspring of you on this picture. For example, scientists like, colleagues like Alan McRae have had students somewhere over here in this supposedly normal distribution. And for that, thank you, Robin, for the unlikely times.